In part two of this lecture for chapter 3 on biopsychology, we'll focus on the nervous system. Now, there are a lot of moving parts, no pun intended, to the nervous system. So we'll break it down into sections. Therefore, we'll first focus on the basic components of the neural system and build from there. Now, from individual nerve cells to brain structures, biological processes control everything that we do. So in this section, we'll discuss the structure and functions of nerve cells and how the most important neurotransmitters relate to behavior. So let's get introduced. The nervous system is a communication network in our body that sends messages between sensory receptors, muscles, and glands. The nervous system allows us to perceive the world, respond to stimuli, and generate behavior. The human brain is the most complex organ in the body and serves as the central control center for the entire nervous system. It plays a crucial role in regulating various bodily functions such as breathing, heart rate, and digestion, as well as controlling movement, sensation, and cognitive processes. Without the brain, the body would not be able to function properly. Now, nerve cells, or neurons, are the basic building blocks of the nervous system. There are billions of neurons or nerve cells throughout the body, but most are in the brain. There are also motor and sensory neurons throughout the body, and they contain various components that aid in the process known as the action potential. The cell body contains the nucleus, excuse me, and this nucleus, along with other organelles, are the powerhouse, the control house of the cell. Now, dendrites are the short fibers that receive messages from other neurons, while the axon is that long fiber that transmit messages from the cell body to other neurons or muscles or glands. And then there at the end, you'll see terminal buttons, and those are like small knobs at the end of the axon that are responsible for sending the signal to other neurons. You also see a pinkish overlaying structure along the axon, and this is the myelin sheath. This is what we call the axon covering that feeds neural messages down the axon. Does anyone actually know what the degeneration of this layer on the cell is called? Some people know if they've been exposed to people with multiple sclerosis, as occurs when the immune system attacks the myelin sheath and causes inflammation and lesions. And this makes it difficult for the brain to send signals to the rest of the body. Now, we'll keep mentioning this communication between neurons, but first we need to consider, how does it occur? So this communication process is referred to as the action potential.
In this video, I'm going to explain an action potential. An action potential, or a nerve impulse, causes a movement of ions across the cell membrane of a neuron, similar to a ripple passing along the surface of a pond. The cell membrane of a neuron contains thousands of tiny molecules known as channels. These channels allow either sodium or potassium ions to pass through. Generally, the channels on a neuron are closed, and the membrane is said to be in a resting state. In this state, the charge of the inside of the cell membrane is more negative than the outside. And if you could imagine sticking microelectrode connected to a voltmeter inside this membrane, the reading would show about negative 70 millivolt, meaning that the inside of the neuron is about 70 millivolt less than the outside. The cell membrane of a neuron is polarized because of this electrical difference across the membrane. Now, a nerve impulse starts when pressure or other sensory inputs disturbs a neuron's plasma membrane, to the point that the potential difference reaches a threshold voltage of about negative 55 millivolt. This causes hundreds of sodium channels in that region of the membrane to open briefly. This in turn allows positively charged sodium ions to flow inside the cell membrane. Now the inside of the membrane temporarily becomes more positive than the outside, which in turn causes the voltage to rise. This is called depolarized state. the membrane is now depolarized because the net charge inside the axon changed from negative to positive as sodium ions entered in. As the impulse passes, the potassium channels begin to open, allowing positively charged potassium ions to flow out, which causes inside of the axon to resume a net negative charge. At this point, the voltage would fall. The membrane is now set to be repolarized because, again, it's negatively charged on the inside and positively charged on the outside. Now, this depolarization and repolarization of a neuron membrane is called an action potential. Action potential is just another name for a nerve impulse. After a nerve impulse, some voltage-gated potassium channels remain open, resulting in further movement of potassium out of the cell. As the voltage falls below the reading of the resting state, the membrane is said to be hyperpolarized because it's even more negative than at the beginning. At this stage, neuron is unable to conduct a nerve impulse and it's said to be in the refractory period. The refractory period is a very short period during which the sodium-potassium pump continues to return sodium ions to the outside and potassium ions to the inside of the axon, thus returning the neuron to resting potential. So in summary, an action potential is just a wave of depolarization and repolarization. It's not an electric current. It's just a series of voltage-gated ions channels opened and closed. Thank you for watching. Make sure you subscribe, and hopefully I made it wicked easy to understand.
So again, this action potential is that neurotransmission process of communication between the neurons. And it's an all or nothing process that only sends signals when the threshold is actually met. The cell becomes briefly charged by the net intake of positive ions, and this is the traveling electric charge created when the channels in the cell membrane quickly allow positive ions in, and then more slowly pump the ions out again as the wave moves on. So we'll examine this full action potential step by step and look at it at each stage because that video was obviously a brief kind of overview. So in step 1, the neuron receives signals from other neurons. Some are prompting it to fire, and while others are inhibiting it to fire, or basically telling it not to fire. So we have to ask, when does the cell send the actual action potential? Well, the answer is when the threshold is reached. The action potential will either fire or not fire, kind of like a gun, right? It can jam or it'll shoot. And this is known, as I said earlier, as that all or nothing response. Now, if there is no firing, then there's no effect and no further stimulation would occur. So when the action potential travels from the cell body to the terminal branches, it moves along the axon. And of course, the signal must be transmitted to another cell to continue this communication process. The message must be transmitted across the synapse to reach the next cell. If signals only have one level of intensity, then we have to consider why things occur. Like, why will the tap hurt? Why does a tap hurt less than a punch? So the intensity of a stimulus is encoded by the frequency of action potentials, not by their amplitude. And this means that stronger stimuli will cause more action potentials per unit time, while a weaker stimulus will cause fewer action potentials per unit time. So this doesn't mean that the action potential is stronger or harder. It just means that there's more occurring in that same ratio of time, which is why a punch or a thump would hurt more than a little tap. So the action potential follows this all or nothing law, which states that any subthreshold stimulus will cause nothing, while threshold and superthreshold stimuli will produce full responses of the excitable cell. So again, that explains why a punch would hurt more than a tap, because it activates more pain receptors in the skin, which sends more action potential to the brain, indicating higher level of pain. So again, that was the question to somewhat consider in that case. Now, neurotransmission is the process of transmitting messages from neuron to neuron. It occurs in a specialized space known as the synapse. And in synapses, the knob-like endings, terminal buttons of the axon, sending neuron faces the dendrite or cell body of the receiving neuron across this gapway. And this area is sometimes referred to as a synaptic cleft, synaptic gap, synaptic junction. I mean, it goes by any other name, basically. Now, as far as this communication process goes, what is the message being sent? Well, if you see the little blue knobs at the bottom, then you know the answer is neurotransmitters. So when neurons communicate with each other, they send electrical signals that trigger the release of neurotransmitters from small sacs called those synaptic vesicles. These neurotransmitters then bind to specific receptors on the postsynaptic cell, either exciting or inhibiting its activity. And this process ultimately determines whether or not the postsynaptic neuron will fire an action potential or not. Now, non-transmitted neurotransmitters are taken back up into the sending neuron after stimulating the receptors on the receiving neuron for reuse. And though neurotransmitters are products of the endocrine system, we do introduce them here as they are the chemical byproduct of the action potential. Again, we'll talk more about them in the section on the endocrine system later in the lesson. But neurotransmitters have their own pathways that deliver specific messages, influencing our thoughts and behaviors. Some neurotransmitters have both excitatory and inhibitory receptors, and these include things like acetylcholine and dopamine. So we'll take a look at this video, which by all means is probably the cringiest video I could find, that describes the various functions and types of neurotransmitters. But hopefully it'll leave a lasting impression. I know it has in the past, so for your viewing pleasure.
Neurotransmitters are chemical molecules produced by neurons. They are communication tools. They send signals to other neurons, muscles, or glands. Excitatory, inhibitory, modulatory commands. While signals passing within your neurons are electrical, communication between neurons is chemical. After job completion, neurotransmitters can be degraded or diffused from the receptor. Concentration is faded. There may also be reuptake, wherein the molecule is reabsorbed by the neuron that released it. Cool. First to be discovered was ACH or acetylcholine. ACH helps run your body's maintenance routine. It contracts smooth muscles, an excitatory function, and slows your heart rate down, an inhibitory function. Whether a neurotransmitter inhibits or excites depends only on the target cell receptor types. In skeletal muscle, ACH binds nicotinic receptors, but in the heart, ACH binds muscarinic receptors. In the CNS, it's in interneurons that it's mostly found. In Alzheimer's disease, the brain's ACH levels are down. Now onto the modulatory neurotransmitter dopamine, which runs the brain reward system and is a catecholamine. Cell bodies producing it are largely found in the midbrain, the ventral tegmental area, and substantia nigra are its domain. Dopamine floods synapses when you get something good. It modulates your thoughts, actions, and even your mood. Dopamine also controls your posture and movement, and it promotes wakefulness, so your time is well spent. Low levels in Parkinson's is what makes movement slow, while high levels in schizophrenia make hallucinations show. Norepinephrine is released from the adrenal medulla. It increases your heart rate but makes digestion slower. In the brain it heightens arousal and so preps you for action. So challenges or threats are met with the right reaction. It's the sympathetic nervous system's main neurotransmitter. It's released by fear and stress, like from reading Twitter. It's related to epinephrine, which makes a fight-or-flight response. A neurotransmitter and a hormone which at adrenoreceptors docks. Serotonin is the brain's key monoamine neurotransmitter. Messages within your gut, and to your brain it delivers. It is modulatory, making tweaks to your systems of cognition, emotions, physiology, and metabolism. It regulates mood, sleep, wakefulness, appetite, and aggression, circadian rhythms, body temp, and neuroendocrine function. Low serotonin results in sleepiness, brain fog, and confusion, and low levels are found in people with depression. The main inhibitor, GABA, keeps neuron messages at bay, inhibiting continuation of signals down a neural pathway. Preventing excess signaling, it keeps activity in check, because excess activity would just result in a wreck. GABA removes signal noise, and it supports sleep. Disrupting GABA signaling makes anxiety run deep. GABA's released from both axon terminals and from dendrites, allowing a fine-tuned response to the excitation that it fights. GABA is synthesized from glutamate despite the fact that it's inhibitory while glutamate excites. Glutamate is the brain's most abundant free amino acid, activating neurons so that excitation is granted. NMDA and AMPA are two major glutamate receptors. They are on the receiving neuron and act as collectors. It aids learning by affecting synaptic plasticity, hence updating the brain's connectivity. Oxytocin forms strong bonds of loyalty and trust, makes the bond between mother and child more robust. It stimulates release of milk from mammary glands. During birth, the uterus contracts on its commands. Oxytocin's made and released from the hypothalamus via the pituitary gland to trigger body responses. Nitric oxide is made on demand. It is a soluble gas, not stored in synaptic vesicles. Through membranes it can pass. When released, it acts directly on molecules in the cell. It's involved in memory and learning from what we can tell.
I do hope you enjoyed that video, and perhaps you might learn a little jingle or two to learn those, excuse me, to be able to memorize what some of those neurotransmitters do. Now, obviously, when we learn about neurons, we often examine one neuron at a time, as we did in the prior slides, and we learn its parts and its function. But our billions of neurons exist in a vast and densely interconnected web. One neuron's terminal branches send messages to neighboring dendrites. And these complex webs of interconnected neurons form with experience. Remember this: neurons that fire together, wire together. So these neural networks are activated when needed for action, because again, it's never just a single neuron in occurrence. It is a vast network, and one is connected to many. So let's apply this theoretical to an actual experience in this next video: neurotransmission and action potential.
How does the pain you experience when you burn your hand result so quickly in an action by your muscles? Many animals respond to environmental stimuli using specialized cells called neurons. A stimulus is detected by sensory receptors, and the body responds through motor effectors. These cells working together allow you to respond very quickly to threats. When you touch something hot, heat receptors of a sensory neuron detect the stimuli and send the information of heat to an interneuron in your central nervous system. From there, a motor neuron sends a response from your central nervous system to the skeletal muscles in your arm, causing them to contract and pull your hand away. The fundamental process of neural transmission that underlies this action occurs in all neurons of the body. Neurons transmit this information through changes in the electrical potential of the membrane by the movement of ions across the membrane. An electrochemical gradient governs the movement of these ions, resulting in an electrical impulse. The resting membrane potential in a neuron, when the cell is not firing an impulse, is established by the unequal distribution of sodium ions outside of the cell and potassium ions inside the cell, making the outside of the cell more positively charged compared to the inside. The electrochemical gradient is established and maintained by an enzyme called sodium-potassium ATPase. When a neuron is stimulated, sodium ion channels open and sodium ions flow into the cell. This leads to a change in the electrical potential across the membrane called depolarization. The depolarizing electrical potential travels down the dendrites and over the cell body. Multiple electrical potentials will combine at the axon hillock in a process called summation. If the depolarization is large enough, an action potential is triggered. Action potentials are all or none electrical impulses that maintain their amplitude and strength down the length of the axon. The action potential travels down the axon when the depolarization of an area of membrane causes adjacent voltage-gated sodium ion channels to open. The influx of sodium ions results in membrane depolarization along the membrane. After a short delay, potassium ion channels open and potassium ions flow out, repolarizing the membrane. For the neuron to fire again, the resting membrane potential needs to be re-established. Sodium-potassium ATPase is used to move sodium and potassium ions against their concentration gradients, re-establishing the resting membrane potential. As the action potential moves down the axon, ions are diffusing only a short distance, allowing the signal to move quickly. At the axon terminal, the electrical impulse passes to another cell at a cellular connection called a synapse. The space between the presynaptic neuron and a postsynaptic cell is called the synaptic cleft. The presynaptic neuron contains signal molecules called neurotransmitters that are packaged inside vesicles. When an action potential reaches the end of a neuron, neurotransmitters are released by exocytosis from the neuron into the synaptic cleft. Neurotransmitters bind to the adjacent cell at receptor sites attached to ion channels. The channels open, allowing the movement of ions into or out of the effector cell, which alters its membrane potential, thereby transmitting the signal from the neuron to the effector cell. Because nerve impulses move very rapidly down the axon of a neuron and move from cell to cell across synapses, you react quickly to a stimulus, like burning your finger.
So we've mentioned that neurotransmitters are the natural chemical messages of the action potential process. However, this process can be disrupted when other substances are introduced to the body.
Most people will take a pill, receive an injection, or otherwise take some kind of medicine during their lives. But most of us don't know anything about how these substances actually work. How can various compounds impact the way we physically feel, think, and even behave? For the most part, this depends on how a drug alters the communication between cells in the brain. There are a number of different ways that can happen. But before it gets into the brain, any drug must first reach the bloodstream on a journey that can take anywhere from seconds to hours, depending on factors like how it's administered. The slowest method is to take a drug orally, because it must be absorbed by our digestive system before it takes effect. Inhaling a drug gets it into the bloodstream faster, and injecting a drug intravenously works quickly too, because it pumps the chemicals directly into the blood. Once there, the drug quickly reaches the gates of its destination, the brain. The entrance to this organ is guarded by the blood-brain barrier, which separates blood from the nervous system to keep potentially dangerous substances out. So all drugs must have a specific chemical composition, which gives them the key to unlock this barrier and pass through. Once inside, drugs start to interfere with the brain's normal functioning by targeting its web of neurons and synapses. Neurons are brain cells that have a nucleus, dendrites, and an axon. Synapses are structures placed along the dendrites or the axon, which allow the exchange of electrochemical signals between neurons. Those signals take the form of chemicals called neurotransmitters. Each neurotransmitter plays different roles in regulating our behaviors, emotions, and cognition, but they all work in one of two ways. They can either inhibit the receiving neuron, limiting its activity, or excite it, creating a new electrochemical signal that spreads throughout the network. Any leftover neurotransmitter usually gets degraded or reabsorbed into the transmitting neuron. A drug's effectiveness stems from its ability to manipulate these synaptic transmissions at different phases of the process. That results in an increase or a decrease in the amount of neurotransmitters being spread. For instance, common antidepressants, like SSRIs, stop the reabsorption of serotonin, a neurotransmitter that modulates our moods. This effectively pushes more of it into the neural network. Meanwhile, painkillers, like morphine, raise levels of serotonin and noradrenaline, which regulate energy, arousal, alertness, and pleasure. Those same neurotransmitters also affect endorphin receptors, reducing pain perception. And tranquilizers work by increasing the production of GABA to inhibit neural activity, putting the person in a relaxed or sedated state. What about illegal or illicit drugs? These have powerful impacts on the brain that we're still trying to understand. Crystal meth, and amphetamine, induces a long-lasting release of dopamine, a neurotransmitter linked with the perception of reward and pleasure. It also activates noradrenaline receptors, which increases the heart rate, dilates pupils, and triggers the body's fight or flight response. Cocaine blocks the reuptake of dopamine and serotonin, pushing more into the network where they boost energy, create feelings of euphoria, and suppress appetites. And hallucinogenic drugs have some of the most puzzling effects. Substances like LSD, mescaline, and DMT all block the release of serotonin, which regulates mood and impulsivity. They also have an impact on the neural circuits involved in perception, learning, and behavioral regulation, which may explain why these drugs have such powerful impacts. Even if some of these effects sound exciting, there are reasons why some of these drugs are highly controlled and often illegal. Drugs have the power to alter the brain's chemistry, and repeated use can permanently rewire the neural networks that support our ability to think, make decisions, learn, and remember things. There's a lot we still don't know about drugs and their effects, both the good and the bad. But those we do know about are the ones we've studied closely and turned into effective medicines. As our knowledge grows about drugs and the brain, the possibilities will also increase for treating the many medical problems that puzzle researchers today.
So our takeaway is that we understand that drugs interfere with how neurons send, receive, and process signals via transmitters. Some drugs, such as cocaine, meth, and heroin, can activate neurons because their chemical structures mimic that of a natural neurotransmitter in the body. And this allows drugs to attach to and activate the neurons. Although these drugs mimic the brain's own chemicals, they don't activate neurons in the same way as the natural neurotransmitter. And this actually leads to abnormal messages being sent to the network. And there are medications that inhibit this reuptake process and help ensure the signals get transmitted. For instance, some people take SSRIs to help reduce depression, and this is done by increasing serotonin levels at the synapse. And most ADHD medications, such as Ritalin, work by blocking the transport of dopamine back into the sending neuron.